Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. So it is the part 2 video of the chapter Heredity and Variation. In my last video I have discussed till monohybrid cross. Today I will discuss rest of the part of this chapter. So this is the diagrammatic representation of monohybrid cross, and this experiment was carried out by Mendel, okay, by observing a single character. Next, Mendel derived some laws or principles after doing his experiment. At first, what is law of dominance? So according to Mendel, when two pure parent, pure, that means pure homozygous parent, cross with each other, they produce a hybrid in F1 generation. The character which is expressed in F1 generation is called as dominant character, and that character which is suppressed by the dominant gene is called as recessive gene or recessive character. Okay, but in F2 generation the recessive character again appear with the dominant character in 3:1 ratio. Okay, this is simply the law of dominance, but there are certain deviations from this law which is called as incomplete dominance. Here in incomplete dominance, none of the character of parent are fully expressed themselves. So in F1 generation, an intermediate character is expressed. For example, when a pure red flower plant is crossed with white flower plant in F1 generation, flower with pink flower plant is produced. So none of the character is produced in F1 generation. That condition is called as incomplete dominance, when none of the parent can express themselves in F1 generation. It is called incompleteness. Next is codominance. Codominance means, unlike dominance, in codominance both allele is expressed, both allele express themselves. For example, AB blood group. So for A blood group, IA is responsible. For B blood group, IB is responsible. But in AB blood group, both IA and IB express themselves. So both of the allele express themselves and coexist in the case of codominance. Okay. Next deviation is multiple allele. So alleles are the alternative forms of a gene. In diploid organism, two copies of a particular gene occur, one each on homologous chromosome occupying the same loci or same loci, same location. The alternative forms of gene arise due to mutation in original. In some cases, there may not be any alternative form. Okay. There is mutation completely eliminates a gene called as null mutation. Sometimes the mutation may not have any effect at all, called as silent mutation. Okay, but sometimes there are multiple forms are present, multiple forms, more than 2 or 3 alleles are present for a given gene, called as multiple allele. Okay, generally in diploid organism, 2 alternative forms are present, one for tallness, one for dwarfness. It is 2 alternative forms, but sometimes more than 2 forms are present, called as multiple allelism. Okay. For example, human blood group, ABO blood grouping system. Here multiple alleles are present. Next, what is the law of segregation? One law is law of dominance. Second one is law of segregation. What is the meaning of law of segregation? What is segregation? So, in diploid organism, two factors of a given character remain together without mixing. That means TT. They present together but without mixing each other. At the time of gamete formation, The two factor or allele segregate from each other. It produce small t, it can produce capital T separately. Okay. And gamete receive only one of two factor of any character randomly as per the principle of probability. Okay, this is called the law of segregation. That means in diploid organism, two allele present togetherly, but during gamete formation they segregate from each other. They are pure, they segregate from each other. So it is otherwise called as law of purity of gamete. Okay. Next, Mendel performed dihybrid cross. In case of dihybrid cross, two pairs of alternative characters are considered for this study. Two traits of or characters such as shape of the seed and another one is color of the seed was considered together to understand this dihybrid cross. Di means two. In case of monohybrid cross, Mendel used a single character, observed a single character, but here in dihybrid cross, he observed two characters. One is shape of the seed, second one is color of the seed. Okay. In these two pair of alternative characters, round seed is dominant over the wrinkled seed, which is smaller. Okay. And yellow seed, that is capital I, is dominant over green seed, that is small y. Okay. Like monohybrid cross, Mendel selected pure breeding plants for both the traits for the cross. So true breeding plant having round and yellow seed is crossed with green and wrinkled seed. This is yellow and round, and this is green and wrinkled seed. Okay. Now he crossed them, pure parent plant crossed with each other to get F1 generation with yellow and round seed because they are dominant characters. Now by selfing, F2 generation is produced. In F2 generation, all the characters are produced in 9:3:3:1 character. In F2 generation, yellow round, round green, wrinkled yellow, wrinkled green, all seeds are produced in 9:3:3:1 ratio. Okay. Now, Mendel derived law of independent assortment after dihybrid cross. So, according to law of independent assortment, it states that in a cross between parents with two or more contrasting character, yellow, green, round, wrinkled, the inheritance of one pair of contrasting character is independent of the other pair. The two characters are transmitted independently. So the two factor for seed color, capital Y and capital Y, and two factor for the shape of the seed, that is round and wrinkled, present together in F1 and assert independently or randomly during gamete formation, so that gamete contains only one of the two factors of a character. As a result, four types of gametes are formed, that is round with yellow, then round with green, round with green, then wrinkled with yellow, then wrinkled with green. Okay, these four type of gametes unite randomly as per probability to give rise to four types of F2 offspring. Hence, inheritance of two or more factor or gene independent of each other. However, it is now well established that genes or factor located very close to one another on same chromosome are called linked and not assert independently. There are some genes which do not show this independent assortment. They are called linked. They are linked with each other because they are present on a same chromosome togetherly, side by side. So they cannot assert independently. But in this case, this gamete can pair with this gamete can pair with this, so they are independent of each other, and they can integrate with any of the gamete by chance, by probability. Okay, so it is called the principle of independent assortment. Okay, next, what is test cross, what is back cross? So back cross is basically the cross between F1 hybrid with any of the homozygous parent. When F1 hybrid is crossed with homozygous dominant parent, then all offspring will be with dominant phenotype. When F1 hybrid is crossed with recessive parent, both dominant and recessive phenotype appear in equal proportion. Okay, as you can see from this diagram. Okay, next one is what is test cross. It is a special kind of back cross when the F1 hybrid is crossed with the recessive parent. Okay, Mendel during his hybridization experiment made several interesting observations regarding the color of various plant component. For example, he noticed that plant with colored seed coat always had colored flower, colored leaf axil. He also observed that pea plant with colorless seed coat had always white flower and no pigmentation in their axil. So Mendel gave no explanation for those observations, but his results indicated that a single gene affect more than one trait or character. The phenomenon of a single gene contributing to multiple phenotypic traits is called pleiotropy. In Mendel's pea plant, seed coat color was not only responsible for seed coat color but also for flower as well as axil pigmentation. Pleiotropy. Pleio means many in Greek and tropic means affecting. So one gene affecting multiple apparently unrelated phenotypic character is called pleiotropic gene. Next, what is polygenic inheritance? Polygenic means Mendel's law of inheritance gives us basic ideas about inheritance from parent to offspring. But this inheritance refers to qualitative character, that is, traits which are easily classified into distinct phenotypic categories. Okay. For example, we find Mendelian experiment how many of the plant become tall and dwarf and how many had yellow cotyledon or green cotyledon. The number refer to qualitative trait of plant but does not let us know how tall, how much yellow the characters are. Such phenotypic categories are under the control of one or very few genes with little environmental modifications to obscure into separate phenotypic changes but forms a spectrum of phenotypes. Technically speaking, the phenotypic classes exemplify discontinuous variability and spectrum of phenotype illustrate continuous variability. Characters such as grain weight, yield, milk production, egg production are quantitative or metric traits with continuous variability. The basic difference between qualitative and quantitative involves number of genes contributing to the phenotypic variability and the degree to which phenotype are modified by environmental factors. Quantitative traits are generally governed by a large number of genes, each contributing to the trait, and the contribution of each gene is so small to phenotype that the individual effects cannot be detected by Mendelian method. The number of genes affecting a single character or trait are together called as polygene. When many genes contribute to a single character, called as polygenic inheritance. Poly means many. Okay. Next, what is chromosomal theory of inheritance? At first, this theory was first discovered by Walter Sudon and Theodore Boveri, and it is also called as Sudon and Boveri theory. It is a very fundamental theory of genetics that states that chromosomes carries the genetic material. The theory proposed early in 1900 by Walter Sudon, and he told that chromosomes are in pair. Okay, homologous chromosomes separate during meiosis, so that alleles are segregated from each other. Meiotic products have one of each homologous chromosome but not the both. Fertilization restores the pair and genes are located on the chromosome, and they also show some parallelism between chromosome and Mendelian factor. And because Mendel did not use the term gene, he used Mendelian factor. He used factor actually. We called them Mendelian factor. So Sudon and Boveri shows this parallelism between Mendelian factor and chromosome. Alleles of a factor occur in pair. T, capital T, capital T. Chromosome also occur in pair. Similar dissimilar allele separate during gamete formation, and homologous chromosome separate during meiosis. Mendelian factor assert independently. Paired chromosomes separate independently during meiosis, but linked gene in the same chromosome normally do not assert independently. So these are some parallel behavior between chromosome and Mendelian factor. Okay, then what is linkage? Linkage was first discovered by Watteson and Punnett in 1906. However, it was put forward as a regular concept by Morgan after his experiment on Drosophila melanogaster. What is linkage? It is the phenomenon of certain genes or behavior of a certain gene for staying together on a chromosome and during inheritance through several generations without any change. They do not separate from each other because they are linked on chromosome. Certain genes are linked with each other. They do not segregate. They tend to remain with each other. Okay, that is called as linkage. These are the characteristic features of linkage. Okay, next, linkage group. What is linkage group? Linkage group is a linearly arranged group of linked genes. Okay, suppose for example in a fruit fly, what is Drosophila melanogaster has 4 linkage group because they have 4 pairs of chromosome. Human being have 23 linkage group because they have 23 pair of chromosome. Okay. Next, what is crossing over? It is the exchange of genetic material between two homologous chromosomes, and it leads to recombination. When two homologous chromosomes exchange their part with each other, it leads to recombination. Okay, so it is all about your chapter Heredity and Variation, and I hope you liked this whole session. And in my next video I will discuss another chapter of your class 12th. So please stay tuned, hit the like button, share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.